Welcome friends to this new lecture of soil science and technology, and in this lecture we will be trying to finish this soil testing, and then we will be starting the soil organic matter. So, in the last two lectures we have talked about different aspects of soil testing. Obviously, it is a very huge topic, and obviously we cannot cover each and every little bit step of little bit aspects of soil testing. But before wrapping the soil testing, let us discuss some advanced methods of soil testing. You have known that from our last lectures that we have got individual extracted for extracting the specific nutrients. However, there is an advanced method of we call it Mehlich 3 extraction, and Mehlich 3 is basically a combination of different chemicals. So it is a collection of different chemicals, and which helps in extracting not only the phosphorus and potassium but also calcium, magnesium, and micronutrients. So it is a very much comprehensive method for extracting different available nutrients. So, Mehlich 3 extraction basically, Mehlich 3 extracting reagents basically composed of these 0.25 molar of ammonium nitrate, and then 0.015 molar of ammonium fluoride, 0.001 molar of EDTA, 0.2 molar of acetic acid, and 0.013 molar of nitric acid. So, it is basically composed of individual these components, and each individual component has a specific role for extracting these micronutrients and macronutrients. So, this is an advanced method. So, once we extract these Mehlich 3 using this Mehlich 3 extractant, obviously the next step is to measure their concentration. For measuring their concentration, generally nowadays the most advanced method is ICP-OES. The full name of ICP-OES is inductively coupled plasma optical emission spectroscopy. Now, this is an advanced method which basically works on optical emission spectroscopy. Another method which is available also, we call it atomic absorption spectroscopy or AAS. So, there are two methods when generally scientists are using. One is called AAS, another is ICP, but ICP is much more precise, I would say. And so, let us discuss what is ICP. The full name of ICP is basically inductively coupled COUPLED plasma and then optical emission spectroscopy. So, why we are calling it inductively coupled plasma, we will see in the next slide. So, ICP-OE is basically inductively coupled plasma, and they are at least 2 times as hot as flames or furnaces. So, if you remember, in case of flame photometry, the source of excitation is generally the flame, which is generated through LPG gas. However, in case of ICP, we generate the flame or plasma instead, we generate the plasma instead of flame, which is 2 times as hotter. And this is basically argon plasma. And the argon plasma is the result of the flow of argon ions in a very strong localized radio field, which we created around the torch, ICP torch. We call it ICP torch. Now, obviously, we get a 6000 to 10,000 Kelvin of temperature, which is very common, and it is very hot enough to excite most elements, so they can emit the light. Because most of the elements will be energized and they will go to the upper higher energy state, and then they will revert back to their ground state, and in doing so they will emit some energy which will be measured. So, this high temperature is hot enough to prevent the formation of most of the interferences and break down the oxides and eliminate most molecular and spectral interferences. So, this high temperature is very much important for removing any type of interferences that could be possible in other methods. So, the way to do the atomic emission spectroscopy is basically nowadays in advanced countries, in more advanced laboratories, they use this method for measurement of these available nutrients. So, you are seeing here a ICP torch. Now how it works? In the ICP torch you will see some concentrated quartz tubes in concentrated rings. Now generally generally the argon is flow, which is the we call it argon plasma. So argon is flow through a tangential flow, and sample is injected from here, and you can see there are two coils, induction coils and magnetic field. So radio frequency electrical current and associated magnetic field is given. So radio frequency electrical current and associated magnetic field is given, and we generally use a Tesla coil to spark or ionize this argon, and ultimately the argon will ionize to form electrons, and these electrons, acceleration of these electrons will the electrons which are produced from this ionization of argon will be accelerated by this magnetic field, and obviously the energy will be transferred from the electrons to the gas by collision. So the gas is heats up. So basically we are incorporating the argon here, and this argon is moving through these quartz tubes, and while it moving through these quartz tubes, we are ionizing is through a Tesla coil, which is creating electrons, and these electrons basically moving through in high acceleration in the influence of magnetic field, and as a result of that, high energy is transferred when this electron is collided with the gas, and as a result the gas is heats up. So at the top you can see the plasma is generated. So this is an emission region. So energy production of high concentration of both excited atoms and ions. So this is the principle behind this inductively coupled plasma. Ultimately there will be two detectors, and this detector will measure the emitted light intensity, and from there it can measure the concentration of a particular element. So the element which are present in the sample, so we are incorporating that. When these are coming in contact with the plasma, obviously they will be excited and going to the high energy state, and as a result they will emit the energy which will be further measured by the detectors. So this is the principle behind ICP OES. And finally guys, I would like to show you another portable tool generally we use in the field, the soil scientists generally use in the field. We call it soil testing kit. Now it is a portable kit which enable farmers to take it to the fields and take the measurements, and it most common kits helps us to find the pH and primary nutrients, NPK along with organic carbon, and kits can also helps in finding all the nutrients, but it is expensive. So soil sample is first taken using a spade or trowel, and then you will see inside this soil testing kit you will see several types of small tools, starting from cuvettes, starting from test tubes, and then you have to put the soil sample within the test tube, and there will be several chemicals, and you have to add those chemicals directly into the test tube, and then you have to develop a color by mixing these reagents, and this color will be finally compared with the standard color chart which is given within the soil testing kit, and from that you can qualitatively ascertain what is the nutrient status of that field. So this is a very quick and rough method. Obviously this is a qualitative method, it is not a very much accurate quantitative method, but it is very much useful when you are considering on-site evaluation of soil fertility. And from that you do not need to go for soil drying and soil grinding, and there are some scoops are already there which can measure around 2 or 2.5 gram of soils. You can use wet soils directly, and you can and these testing kit can be used for measurement of most of the routine soil tests. So guys, so this is about the soil testing kit. I hope that and these are the references. I hope that you have got a basic overview of soil testing and you got a idea of different aspects of soil testing. Obviously, I didn't cover all the important aspects because the limitation of time. But however, I would expect that you will go ahead and you know uh, consult different books and different scientists for measurement of your measurement of soil samples and other parameters. So let us wrap up here, and these are the references. So in the next topic will be very important, that is soil organic matter and climate change. So let us uh, let us see what is soil organic matter. Obviously, soil organic matter consists of a complex system of substances which is ranged from component of organic residues undergoing decomposition, metabolic products of microbes, and then products of secondary synthesis of humic substances. So this is a complex mixture of different organic fractions. Now, this organic matter, soil organic matter, is basically present in the soil. If you remember, in the first lectures we have talked about that volumetrically it contains about 5% in the soil, but it is very important, as you know, since it has got high amount of catenation capacity as well as high amount of nutrient holding capacity. And so the organic matter or organic matter contains several organic fractions starting from the fresh litters which is present over the soil surface, then decaying organic matter, then a stabilized form of organic matter which is resistant to further decomposition, we call it humus. And remember that the humus is the major fraction of organic matter that is responsible for soil physical, chemical and biological properties. We will discuss the humus in details later on. So basically soil organic matter serve as a soil conditioner, nutrient source, substrates for microbial activity, preserver of the environment and major determinant of substantial agricultural productivity. Why? We will see. So if we consider the global carbon cycle, obviously the carbon is recycled within these major reservoirs. You can see soil, then ocean and lakes, then carbonate rocks, then fossil fuels, then atmosphere, vegetation. These are some big pools of or major pools of carbon in the earth and surrounding atmosphere. And you can see that how this movement of carbon is maintained from one pool to another pool. Obviously the global carbon cycle, you can see here the numbers which you are seeing inside the box are basically indicating the pentagrams of carbon stored in that major pool. So in case of soil, obviously 3 to 4,000 pentagrams of carbon stored in carbons. So the number by arrow shows the amount of carbon annually flowing in pentagram per year by various processes between these pools. So also you have to note down that the soil contains almost twice as much as carbon as the vegetation and the atmosphere combined. So this is very important. So you can have a basic idea about how this carbon is moving from one pool to another pool, and it is a very huge amount. And obviously soil is considered as one of the major important pool of carbon, global carbon cycle, which contains twice as much as carbon as the vegetation and the atmosphere combined. So let us see what is organic matter decomposition in soil. So decomposition basically involves the breakdown of large organic molecules into smaller, simpler components. Plant residues are the principal components undergoing decomposition in the soil. So the plant residues are the primary source of soil organic matter. Obviously plants are present in huge amount in the over the soil surface, so they are the primary source of soil organic matter. Now, the rate of decomposition depends on the composition of the plant residues and the soil environment and moisture content in the soil. So, decomposition process can be divided into two major categories. One is aerobic decomposition, another is anaerobic decomposition. Now, if you consider the organic matter composition in the soil, decomposition of the soil, obviously you have to talk about the composition of plant residue, because this is one of the major indicator of how these different fractions of organic organic matter will undergo decomposition. So, volumetrically you can see there are composition of plant residues. Obviously you can see there are 75% of water here, whereas dry matter is only 25%. So, if you see the dry matter, obviously the dry matter contains 42% of carbon, and then 8% of hydrogen, and then 8% of ash, and oxygen 42%. Or in other words, if we concerned about the type of compounds, obviously you see the majority is cellulose, which is the 45%, followed by lignin, that is 20%, hemicellulose 18%, polyphenols 2%, fats and waxes 2%, and sugars and starches 5%, and proteins 8%. So, these are the chemical composition or biochemical composition of the plant residues, and obviously depending on that organic matter decomposition or rate of organic matter decomposition varies obviously. So, rate of decomposition obviously depends on these factors. For example, you can see that in the plant residues, obviously there are sugar, starch and simple proteins, crude proteins, hemicellulose, cellulose, fats and waxes, and lignin and phenolic compounds are there. Remember that these sugar, starches and simple proteins are generally undergo rapid decomposition, and as we go from the top to bottom, obviously their resistance to decomposition increases. So, lignins and phenolic compounds are the most resistant to decomposition, and followed by fats and waxes, cellulose and hemicellulose. So, these are very important aspects you have to take care of, you have to consider. And so, the rate of decomposition obviously depends on the composition of the particular organic matter. So, let us talk about the decomposition of aerobic soil in aerobic soil. So, plant carbon compounds are enzymatically oxidized, as you can see in this reaction. It is enzymatically oxidized to produce carbon dioxide and then water and energy, huge amount of energy, and decomposer biomass obviously. So, essential nutrients such as nitrogen, phosphorus, sulfur are released and are immobilized by a series of specific reactions that are relatively unique to that element. We have already discussed about those in our previous lectures. So, new compounds are obviously synthesized by microbes as cellular components or as a breakdown products or secondary metabolites, and some of the original plant compounds, their breakdown products and microbial compounds become physically or chemically protected from further microbial decay and via interaction with the soil environment. So, these are some important aspects or important dynamics which occur in the aerobic decomposition of soil, aerobic decomposition of organic matter. So, again here this is an organic matter, obviously we are aerobically decomposed in the presence of enzymes and producing carbon dioxide, water, energy and biomass of the decomposers. So, let us talk about decomposition in anaerobic soil. Obviously, this decomposition process is slower than that of aerobic soil, and it generally you can see this type of decomposition in wet and anaerobic condition, and because of this slow decomposition in this anaerobic process, this type of soil, that is wet and anaerobic soil, tends to accumulate large amount of partially decomposed organic matter. Because of mainly two reasons: under low oxygen anaerobic condition, the decomposition takes place much more slowly than that when the oxygen is plentiful, and secondly, certain products of anaerobic metabolism are toxic to many microbes and acting as a preservative for organic matter. So, the end product still contains the energy. So that's why alcohol and methane, which are the major end products of this anaerobic decomposition, are produced, are used as a fuel. But remember that some products are toxic to the plant growth and contributed to the climate change, specifically the methane, which is a very important greenhouse gas. Now, the following reaction you can see here, first of all, propionate is reacting with in the presence of bacteria producing acetate, carbon and methane in the anaerobic decomposition. This is again important, and here you can see acetic acid is anaerobically decomposed to produce the methane, and this carbon dioxide is again reacting with hydrogen to produce the methane. So, this methane is a very notorious greenhouse gas, and which increases the greenhouse gas effect in our earth. So, obviously this anaerobic decomposition is not environmental friendly per se, but this methane can be also used for a as an energy source. So, what are the factors? Obviously, the bacteria which generates methane are called methanogenic bacteria, and remember that there are several methanogenic bacteria or methanogens which plays a key role in this anaerobic decomposition of organic matter. So, let us see what are the factors which controls the rate of decomposition. Obviously, there are several factors starting from temperature, then moisture, then CN ratio of the plant residues, then availability of nitrogen in the soil, and then soil ecology and quality of litter, lignin and polyphenol content. All these are very important as far as the rate of decomposition is concerned. So, in this graph you can see there is a relation between time after planting residues on soils in days and at the biomass remaining. So, you can see here there are three different crops here. So, first of all, this line, this solid line is basically representing rye, and this green is basically represented by vetch, and in case of vetch, obviously the CN ratio is very only 10, and in case of rye it is 87. So, obviously from our previous lecture we know that when there is a when the CN ratio is low, obviously the mineralization is encouraging, it encourages the mineralization, and that is why you can see the decomposition of organic matter is quite rapid in case of vetch as compared to the rye, because in case of rye the CN ratio is quite wide, and as a result mineralization and decomposition is quite slow. And when we mix the 50% vetch with 50% of rye, obviously you can see some intermediate trends. So, that shows that the CN ratio of the plant residue is very important, which controls the rate of decomposition of organic matter. So, another important is quality of organic matter. Obviously, you can see here this portion is basically indicating the net mineralization, and this is net immobilization. So, here you can see different graphs showing decomposition rate or mineralization and immobilization rate for different components. So, you can see here different composition. So, you can see here these basically shows low lignin and polyphenol and low CN ratio. So, that is the ideal condition, because you know that lignin and polyphenols are resistant to decomposition, and when we have low CN ratio, obviously it will also encourage the mineralization. So, obviously we are getting high amount of mineralization. However, when there is a high lignin and polyphenols and low CN ratio, obviously the increasing trend is we are getting, but it is somewhat low because of the presence of high lignin content. Now, in case of high lignin and polyphenol and high high CN ratio, obviously there will be net immobilization, and low lignin and polyphenol and high CN ratio, obviously that will also go with net immobilization. So, that shows the quality of organic matter is a very important determinant of rate of decomposition. Alright. So, Now you see how organic matter basically forms. So this is a very good representation of formation of organic matter. So let us see how it goes. So for describing this slide, I will go back and forth with the next slide. So let us see. So the microbial biomass, or upper right, as you can see, this is upper right, the microbial biomass produces enzyme that break down plant residues. So these microbial biomass basically produces, you can see, enzymes which basically decompose all the plant residues. So plant contain different types of components, as you know that cellulose, hemicellulose, starch, sugars, lignin. So basically they decompose all the components, these enzymes. And when these compounds are metabolized by microbes to obtain the energy, most of their carbon is released as carbon dioxide, while the other element may be released into the soil solution. Now we already we have already seen in case of aerobic decomposition that carbon dioxide is generating due to this enzymatic oxidation, and after carbon dioxide is released, the nitrogen, sulfur and phosphate, they are already they dissolve and also released. So may be released into the soil solution. So the labile carbon, that is upper half of the diagram. So this is the labile carbon and this is the humus or stabilized form of carbon. Remember that this labile carbon is subjected to rapid metabolism. Some components become stabilized. So this labile carbon becomes rapid decomposed, and some component becomes stabilized and goes to this pool, that is humus, and bits of plant tissue and microbial cell, that is particulate organic matter, may become inaccessible to microbial attack and microaggregates from around them. So obviously some dissolved organic matter and plant tissue, particulate organic matter, they are protected by inaccessible, inside the microaggregates and becomes inaccessible. So they move from this labile pool to the stabilized pool. And microbial oxidation creates zone of polarity in formerly hydrophobic biomolecules, allowing their carbon to become protected by bonding to mineral surfaces. So you can see protecting by sorption to the mineral surfaces, obviously. So this is another way. So this is the whole dynamics of soil organic matter that goes on within the ecosystem, and you can see the labile pools is this fraction. Basically this fraction is basically the labile pool, and this is the more stabilized pool or humus, which is responsible for soil physical, chemical and biological activities. Okay. So if we see now if we divide the soil organic matter into labile pool and slow to decompose pool or stabilized pool, obviously this labile pool shows rapid turnover of carbon or labile organic matter, whereas this slow to decompose pool shows protected or slow turnover of carbon. We know that humus is the protected or slow turnover of carbon. Humus is a kind of stabilized product. So for this labile pool, obviously we got some living organism biomass, and then free identifiable dead tissues and detritus, we call it detritus, and then free bits of partially degraded tissues or particulate organic matter, and then free or dissolved biomolecules and degradation products. So all these comes into the labile pool, and they can show rapid decomposition and changes. However, in case of humus, obviously you can see protected bits of degraded cell walls and tissue, that is particulate organic matter, and protected biomolecules or supramolecules and degradation products, and black aromatic products of fire, we call it char. So these type of compounds and can be seen within a soil organic matter. So obviously soil organic matter, again we can divide it to labile pool and non-labile pool or stabilized pool, and labile pool goes rapid decomposition or rapid changes. However, stabilized pool is more or less stable. Humus is important component in the stabilized pool, and in case of labile pool, living organism, free identifiable detritus tissues and particulate organic matter, all these represent the labile pool. So guys, we have covered this this slide up to let us wrap up here. in this lecture, and in the next lecture we will be trying to finish this this organic matter, and then we will be discussing about different organisms which are present in the soil. So, thank you, and let's finish this in the next lecture.